Hi, this is Dr. Crawford bringing you from Dr. Crawford, What's New in Cancer Research. Today I want to talk to you a little bit about a meeting that I attended in mid-November at the Dana-Farber Cancer Institute in Boston on the BRCA gene mutations and latest developments in our understanding of their role in the development of several types of cancer. So the focus of this meeting was the occurrence of mutations in two genes called BRCA1 and BRCA2. And mutations in these genes are implicated in several important types of cancer, including breast cancer, prostate cancer, ovarian cancer, and pancreatic cancer. And these are called germline mutations. In other words, many people who have cancers associated with the BRCA genes have inherited a defective form of the gene. So we're going to talk about a little bit about how that relates to the development of cancer with respect to genetics, as well as to the pathophysiology associated with the onset of malignant transformation. Now, for a long time, we've known that inherited defective alleles of BRCA1 and BRCA2 are implicated in breast cancer. And this particular recording that I'm making today is not intended to go into depth on these types of mutations, but rather just to summarize some of the latest findings that were presented at this research symposium. New research into the role of these BRCA mutations in breast cancer has involved techniques associated with taking a single cell and being able to sequence the nucleic acid, determine the genes that are present and active in particular in the individual cells, both from normal tissue and breast cancer tissue, and tissue that may have an inherited mutation in the BRCA gene, to give us a valuable insight into early stage development of cancer. And this has been made possible by the ability to dig deep into single cell nucleic acid sequencing methods. And it's allowed researchers to document changes that can occur in the breast prior to the development of overt breast cancer. Now, just as a brief summary, BRCA genes. So we have the BRCA genes are located on separate chromosomes in the human genome. BRCA1 is located on chromosome 17, and here's its approximate location on the long arm of 17. And here we have BRCA2, which is located on the long arm or the Q arm of chromosome 13. So numerous mutations have been identified within each of these two genes, and individuals who develop breast cancer as a consequence of these inherited mutations in either BRCA1 or BRCA2, meaning that we have two copies of every gene, and in these patients, they inherit one copy of the gene as normal and the other copy is abnormal. And it turns out that over time, what happens is that a second mutation occurs in the one remaining normal copy of the gene, and this leads to the development of cancer. So it's an inherited predisposition. So over time, in the breast tissue, a second mutation occurs so that neither copy of the BRCA gene is functioning. And this contributes directly to the occurrence of breast cancer. Mutations in the BRCA gene, even in carriers with only one copy, are associated with genetic instability and changes within normal tissue that precedes the development of the malignant tissue and precedes the mutation, the occurrence of the mutation in the second copy of the gene. And these types of changes include increased copy numbers of DNA segments, as well as unusual combinations of chromosomes within the incipient abnormal tissue. So here we see each of the two chromosomes associated with BRCA mutations. BRCA1 on chromosome 17. Here's the abnormal copy, the normal copy. And BRCA2 on 13. Here's the abnormal copy and the normal copy. These abnormal alleles can be inherited either from the mother or the father. And you may have either a BRCA1 or a BRCA2, what's called a germline mutation. What happens over time, however, is that the second copy of the gene becomes inactivated in cells that ultimately become malignant tumors. So recent research results looking at RNA sequencing of individual cells from precancerous normal tissue from patients have shown that in many of these patients who have inherited a BRCA gene mutation, there is a duplication in the Q arm of the long arm of chromosome 1, and also a deletion in the Q arm of chromosome 10. And these precancerous mutations occur in patients who will later go on to develop breast cancer. But at the time the mutations are detected, they're still in the normal tissue. So you can see how these types of studies are facilitating our understanding of early changes in tissues that precedes the development of malignancy. And this is a new window of opportunity to look into early stage changes in normal tissue that precede the development of cancer in a way that has never been possible before. These changes are observed in the luminal cancer precursors, and these represent a common set of alterations. So they're seen in many patients that precede what is called the loss of heterozygosity, meaning the loss of the second copy of the BRCA gene. So these unusual deletions on chromosome 1, for example, and chromosome 10, these precede the loss of heterozygosity. So they're fairly early events, and therefore they're associated with having only one abnormal copy of the gene, and that seems to be sufficient to predispose to these types of early genetic changes. Since these changes occur before breast cancer develops, they can be thought of as predisposing alterations. But why do they occur at all? And current thinking is that this pattern of changes precedes the loss of heterozygosity. We know that's true. And they are not random changes because we see them repeated among patients who have different overall family histories, unrelated patients. And there are some non-specific changes that are observed too. So it's a pattern of genetic change that is associated with the loss of a single allele of the BRCA gene. And it seems to perhaps be associated with a form of biological selection, which is an aneuploidy or an abnormality in the chromosomal content of the cell, which increases the fitness of cells so that they can survive once the second copy of the BRCA gene is lost. So that all of this represents an evolutionary pathway to the development of cancer. And it's related to the loss of function of the original single allele, which may impact genetic repair processes within the cell. So the genetic instability that occurs across the entire genome that has been mapped and studied and sequenced in individual cells has been defined as extreme aneuploidy because there's so many changes that are documented. Some, again, are random. Some are common among patients who go on to develop breast cancer. And they're apparently related in some way to the loss of the BRCA allele in individuals who inherit this germline mutation because they're not seen in individuals who do not inherit this mutation. Importantly, there is a characteristic fusion that's been observed between chromosomes 1 and 16, and it's a recurrent event among large groups of patients. And again, it appears to be a selection-based event and may have an important role in development of breast cancer. So it's very important to determine what's at the basis of this fusion and how does it impact the development of cancer. Now on this panel, what I have here is just, let me see if I can move this a little bit, just showing you some of the changes that can occur as a consequence of the genome instability associated with BRCA mutations. So here we have a hypothetical gene, a set of genes on a single chromosome, genes A, B, C, D, E, F. So a common event that's seen in patients who have the abnormal allele, germline mutation, is what we call a copy number variant. So you see here that segment C is copied several times over, and that's due to errors in DNA during recombination. For example, this can happen, and usually the BRCA gene product will repair these, but if it's unrepaired, you're going to start accumulating these BRCA copy number variants. Here we have a whole segment duplication. So whereas we should have A, B, C, D, and E, now we have A, B, C, D, and E, and C, F, G, C. So there's whole segments here that have been duplicated, and again, due to usually a recombination repair errors. This is an example of a complex copy number variant, and so you can see the extended pieces here that are present in duplicate and in the wrong order. And this is an inversion where a piece, you get a break in, 2 breaks in a chromosome, the piece inverts before it's repaired. And so instead of A, B, C, D, E, F, we have A, B, C, F, E, D. And now we have a translocation, which could be similar to what we're noticing in chromosome 1 and 16, where a piece from one chromosome gets appended to a piece on another chromosome. And that then results in, can result in abnormalities of gene regulation and expression that could impact the development of cancer. So these varied types of mutations are associated with the fact that the germline mutation in BRCA is causing the failure of normal DNA repair processes to occur, even though one copy remains functional. So I'm just trying to get on the right page, which I'm seemingly having a problem with. I'm just going back. I don't know where I ended up here. Okay. Now, in addition to seeing these types of abnormalities in breast cancer, we also see them in certain other types of cancer, for example, in certain types of prostate cancer. So there are many different types of prostate cancer, and at some point I'll be going into greater depth into this type of cancer. But for the purposes of today's summary, suffice it to say that there is one subgroup of prostate cancer that's associated with mutations in the BRCA genes. And this represents a subset of prostate cancers that tends to be quite serious. So we know there are all different types of prostate cancer, and some of these are treatable, and individuals can live long and healthy lives despite the occurrence of this disease. This type of mutation, however, is associated with a more serious form. The BRCA mutations, the germline mutations implicated in prostate cancer are associated with a more serious form of this disease. So what happens is a de novo metastatic prostate cancer will often appear. So instead of the slowly growing, more less invasive type of tumor, we have the rapid appearance of a prostate tumor that shows metastatic properties. And that's frequently associated, when you look at the genes, with a BRCA2 mutation. And this is a very important subtype of prostate cancer under intensive research because the mortality rate associated with inheriting this defective copy of the BRCA2 gene is significantly higher than for other types of prostate cancer. Likewise, there are germline mutations in BRCA that are also linked to ovarian cancer. So evolutionary changes of the BRCA genes are implicated in some forms of ovarian cancer. For example, 20% of patients with high-grade serous ovarian cancer have mutations in the BRCA gene. And again, these mutations are associated with defects in DNA repair, which is a necessary component of recombination, which is a fundamental process that occurs during meiosis and even can occur in mitosis. So this is a very serious form of ovarian cancer linked to the germline mutation in BRCA. And again, it's currently being very heavily researched, but we see again that very invasive, difficult to treat form of ovarian cancer linked to an inherited mutation in BRCA that ultimately results in the complete loss of function of this gene in tumor tissue. Now, again, the changes that are observed in ovarian cancer as well as in breast cancer and other types of cancer seem to be dependent on the genomic instability associated with preliminary stages of disease development. And these are all occurring prior to the onset of overt cancer. So the genomic instability seems to be linked very much to the loss of heterozygosity, the loss of a single copy of the BRCA gene, conferring a less rigorous process of DNA scrutiny with respect to repair mechanisms, and therefore leading to the occurrence of accumulated mutations and genomic instability that ultimately causes disease progression. And that instability is then ultimately linked to the loss of heterozygosity in BRCA, which then is very much linked to the development of a full-blown malignancy. In the case of ovarian cancer, this genetic instability has been linked to, specifically, a loss in an HLA allele on the short arm of chromosome 6. And the HLA allele is important in immune system recognition, and the loss of that allele results in immune escape, and therefore becomes a positive selection factor for the development of ovarian cancer. So it's interesting to see how these early events, loss of an allele, an HLA allele, before the tissue is ever malignant. It's in normal tissue. So you see how the tumor, we're studying and learning so much more about how tumors develop, that this type of study provides concrete evidence that a downregulation of immune system recognition is a critical component of the development of cancer, and that these genetic changes precede and are required to precede the emergence of full-blown malignancy. So these studies at the level of single-cell RNA sequencing are providing tantalizing insights into early changes in immune system recognition patterns in pre-malignant tissue that may ultimately provide a selection signal facilitating the development of malignant tumors that are then resistant to immune surveillance. So we're learning so much more, and it's going to open the door to so many more opportunities for cancer prevention. So interestingly, the patterns of alteration in the BRCA-associated ovarian cancer development are linked to the changes, as I mentioned, in the immune surveillance mechanisms. And this has been revealed by multi-site disease collection. So you can take normal tissue and tissue at various stages of pre-malignant transition to see what's going on with respect to the genes associated with immune surveillance that play an important role in preventing the development of cancer. And what has been seen by looking at these single-cell, multi-site DNA sequence analyses and RNA sequence, importantly, in order to obtain the gene expression profile, some of these mutations are expressed in the form of recombination deficits, again, resulting in duplications and deletions, as well as changes in the immune system recognition. So again, there's this microcosm of selection that is facilitating the outgrowth of cells that have escaped immune surveillance, that have a growth advantage, and that are capable of surviving under conditions where we're going to lose an important DNA repair mechanism once that loss of heterozygosity occurs. So without these additional concomitant changes, we would not see the development of a cancer. In fact, instead we would see cell death. BRCA mutations are also associated with some forms of pancreatic cancer. Okay, so again, we see a very serious cancer and certain forms, not all, but some pancreatic cancers are associated with the germline mutations in BRCA1 and BRCA2. And again, representing an unstable subtype associated with DNA damage repair deficiencies. And in this particular case, what's been noticed, and in certain other cancers as well, that these types of repair deficiency are associated with increased sensitivity to chemotherapy regimens that are involved in damaging DNA, because the cell is already disabled in repairing based on the BRCA presence of the BRCA mutation. So repair mechanisms are disabled to begin with, and then adding a drug that induces further damage to the DNA may preferentially select for the demise of these tumor cells. So this is kind of an explanation of the molecular basis of sensitivity to these types of chemotherapies. Of course, interestingly enough, what we do see is that over time, resistance to these chemotherapy approaches can occur because of the fact that you get repair mutations in the repair genes. You get reversion. So the survival mode is the key driver here, natural selection at the level of the tumor. So in summary then on the BRCA mutation, given the role of the BRCA genes in DNA repair mechanisms, it's not surprising that some of the genetic instability associated with the development of malignancies in BRCA heterozygotes who have inherited a germline mutation in one of these genes may be linked to differences in activity based on the loss of a single functional allele. So it's not an all or none occurrence. There are phenotypic consequences to losing a single allele as a consequence of inheriting the mutation. And this again is an idea of an example of haplosufficiency, in the sense that you get viability, survival in the context of a single allelic deficit. Nevertheless, there are fundamental consequences, in this case affecting DNA repair mechanisms, that strongly predispose to the development of cancer in certain tissues of the body. With an eye towards breast cancer prevention, we know that BRCA1 mutations are associated with triple-negative breast cancers, which can be more difficult to treat than other types of breast cancer. Those that are estrogen receptor negative, progesterone receptor negative, and HER2/neu negative, the triple negatives, these are more difficult to treat generally, although new approaches have been developed. BRCA2 mutations are more likely to be associated with the far more common estrogen receptor positive, progesterone receptor positive breast cancer. So again, this is sort of a genetic cataloging, which is important for us to understand the relationship between these different types of genes and hormonal status in the development of cancer. So the work presented here by numerous researchers studying breast cancer and ovarian cancer, pancreatic cancer, and prostate cancer from Dana-Farber and Memorial Sloan Kettering and other large cancer institutes. The goal of their research is to understand these early precancerous changes associated with phenotypes that will ultimately result in emergent clones of malignant cells. And this work is providing a great deal of insight into certain patterns and mutations that are currently now being linked to specific changes, for example, in the breast tissue associated with reproducible changes in multiple patients that appear to be linked to the development of disease. So loss and gains of chromosomal segments, various forms of aneuploidy, all seen in histologically normal tissue and whose identity is determined by single cell RNA and DNA sequencing. And these types of observations were heretofore unavailable to assessment. So this is really opening a new door in the history of cancer research. Changes associated with germline BRCA mutations are more prevalent and in luminal than basal tissue. You can see more losses or gains, and all of these gains in terms of the various types of mutations associated with deletions and copy number variants, etc. They all occur, again, just to reemphasize, in the context of a haploid sufficient phenotype, as the loss of heterozygosity has not yet developed. So this really is a new window of opportunity to view these early changes. However, it is known that replication stress in these luminal BRCA plus minus tissues, they're plus minus because one copy of the allele is present, one is defective. These were associated with checkpoint deficits that ultimately culminate in malignant transformation, and that involves growth advantage and immune system loss of recognition by immune system components. Strikingly, the genomic instability associated with the germline BRCA mutations may represent a damage signal that triggers inflammatory responses that may further contribute to disease progression. And that's a very big interest of mine, that there is quite a bit of convergence of many abnormal processes that are of somewhat perhaps disparate, though related origin, in terms of how they impact inflammatory processes and immune system responses that are subverted by the tumor cell to promote tumor survival. And the germline BRCA mutation itself is linked to inflammation because of the effects on DNA damage within the cell. So all of these studies are intriguing as they provide a new insight into the early history of neoplastic disease. So thank you. I am at Southern Connecticut State University, which you may not be aware, and have the Cancer Biology Research Laboratory there. And again, I attended this meeting at the Dana-Farber in November 2023 and reporting the latest findings. Hopefully you found it of interest and importance. Thank you.